Hello, science fans! Today we're diving into chromatography, a clever way scientists separate mixtures to make sure our medicines, food, and water are safe. Imagine a jar of mixed candies, some big, some small, some sticky. Chromatography is how we sort them out, but at the molecular level. Think of it as a race. Different molecules are like animals on a tricky racetrack, each moving at its own speed. Some get stuck, some zip ahead, and by the end, they're all separated. That's the magic of chromatography, using differences in speed to split mixtures into their parts. The word comes from Greek chroma, means color. Graphene means to write. Early experiments separated plant pigments into colorful bands. Today it's used for much more: checking drug purity, finding pollutants, and testing food nutrition. Chromatography is a cornerstone of modern science, all based on this simple race. Scientists rely on it to analyze complex mixtures quickly and accurately. It's not just about colors anymore, it's about safety, quality, and discovery. Whether in a lab or industry, chromatography helps us see what's really inside. It's a powerful tool for understanding and improving the world around us. So next time you take medicine or drink clean water, remember the molecular race that made it possible. Chromatography, simple in concept, essential in practice. Let's see how this race really works. To run our molecular race, we need two key players: the stationary phase and the mobile phase. The stationary phase is the racetrack, solid or liquid but always unmoving. The mobile phase is the river that carries our molecules forward, either a liquid or a gas. The magic happens in how molecules interact with both phases. Some molecules love the stationary phase and move slowly, others prefer the mobile phase and race ahead. This difference in attraction is what separates them. Imagine kids at a playground. Some linger on the swings, others head straight for the exit. By the end, each type is separated based on how much time they spend playing versus moving. Molecules that stick to the stationary phase travel slowly. Those that don't, move quickly. This is the heart of chromatography, separating mixtures by how they interact with their environment. By adjusting the phases, scientists can fine-tune the separation for any mixture. It's a simple idea with powerful results. That's how we get a neat, clear separation of molecules. Now, let's talk about High Performance Liquid Chromatography, HPLC, the Formula One of chromatography. HPLC uses high pressure to push the mobile phase through a column packed with tiny particles, making separations faster and sharper. The result? Complex mixtures are separated in minutes, not hours. It's all about precision and speed, crucial for pharmaceuticals and research. Think of regular chromatography as a lazy river. HPLC is a whitewater rapid with a motor. The high pressure and fine particles mean better separation and quicker results. That's why HPLC is the gold standard in labs worldwide. When accuracy and speed matter, HPLC leads the race. Just as there are different race cars for different tracks, there are different types of HPLC for different molecules. The most common is reverse phase HPLC, a non-polar, oily, stationary phase, and a polar, watery, mobile phase. Polar molecules zip through, non-polar ones stick and lag behind. Normal phase HPLC flips this, a polar stationary phase and a non-polar mobile phase. Great for water-insoluble compounds. For charged molecules, ion exchange HPLC uses a charged stationary phase to separate ions by their attraction. Size exclusion HPLC sorts molecules by size. Big ones exit first, small ones take the long route. Chiral HPLC separates mirror image molecules, vital for drug safety. Each type is tailored for a specific challenge. By choosing the right method, scientists can separate almost anything. That's the versatility of HPLC. No matter the mixture, there's a way to split it. The right tool for every molecular race. An HPLC system is a well-oiled machine, made of a few key parts. First, the solvent reservoir holds the mobile phase, pure, bubble-free liquid. The pump pushes this liquid through the system at high, steady pressure. Next, the injector, or autosampler, introduces the sample into the flow. The column is the racetrack, packed with stationary phase where separation happens. After the column, the detector spots the separated molecules as they exit. The computer turns these signals into a chromatogram, a graph showing what's in the sample. Each part is crucial for accurate, reliable results. Together they make HPLC a powerful tool for analysis. It's teamwork at the molecular level. The column and detector are where the real action happens. The column, a steel tube packed with tiny particles, creates the perfect environment for separation. Smaller particles mean better separation, but require higher pressure. Guard columns protect the main column from damage and contamination. After separation, molecules flow into the detector, the system's eye. The most common is the UV-Vis detector, which measures how much light molecules absorb. Other detectors, like refractive index or fluorescence, handle molecules that don't absorb UV light. For ultimate ID, HPLC can be paired with mass spectrometry, LC-MS. The right detector depends on what you're looking for. Careful column and detector choices mean precise, reliable results. That's how we see the invisible world of molecules. Why all this effort? Because HPLC is essential across science and industry. In pharmaceuticals, it ensures drugs are pure and safe, required by agencies like the FDA. Environmental scientists use it to detect pollutants in water, soil, and air. Food scientists check for vitamins, sugars, and contaminants. Clinicians monitor drug and hormone levels in patients. Forensic experts solve crimes with it. HPLC's power to separate and measure makes it a scientific super tool. It keeps our medicines safe, our food pure, and our environment clean. As technology advances, HPLC only gets better. Chromatography racing molecules for a safer world.